Well, thanks a lot. It is always a pleasure to be here. Lamentablemente, como Mariano, no puedo hablar o dar mi seminario en español porque me sale muchísimo más malo de lo que habitualmente me sale. En inglés son malos, pero en español son peores. Así que tengo que necesariamente hacerlo en inglés, hay muchas palabras que no las sé en español. Desde hace bastantes años, a long time I have been working on malaria. And as you all know, malaria, it is a very complicated infection that it has a lot of different cycles and stages during the parasite infection. And we can identify several of them. And you just look at them and they look completely different. You know, the infected form, the sporozoite, the second infected form, if you will, in the liver stages after they infect hepatocyte, and then the parasite leave hepatocyte and they infect red cells, and this is the symptomatic part of the infection. The antigenic makeup as well as the gene expression of these different stages is completely different. So the immunology of these different stages is obviously very different. We know that antibodies are quite important in terms that they can recognize the surface of the sporozoites, and if they are in enough quantity, they are capable of neutralizing the infectivity of the sporozoite, and they do not infect hepatic cells. However, some of them can escape, and once they infect the liver, or specifically the hepatocyte, this hepatocyte manage to present antigens, which are parasite-derived antigens that present in the surface to CD8 positive T cells. And then this CD8 positive T cell recognize the antigen and can eliminate the infected hepatocyte, thereby stopping the infectivity. And in many cases, if the number of CD8 cells is large enough, they can completely wipe out the infection. After they leave the liver, then they infect the red cells, and in this case, antibodies are the ones that seem to have a predominant protective effect if they are in enough quantity, as I said before, as they can inhibit the invasion and reinvasion cycles of merozoite into the bloodstream. What I'm working, I work in a pre-erythrocytic stages, in these particular stages, and in particular on CD8 cells. That's what I'm going to talk about. The immunology of these different stages is completely different because, as I said before, the antigenic makeup is absolutely distinct. Immunity against these stages do not have much of an effect against these other stages or blood stages, and vice versa. So it's like two different parasites that we work on. Okay, so what do we know about T cell immunity against liver stages? This is a hepatocyte which has been infected by a sporozoite, and this is 36 hours after infection. After a single sporozoite that invades this hepatocyte, in 40 hours or 36 hours, 10,000 new parasites may actually develop inside a single hepatocyte. A fantastic rate of proliferation and differentiation at the same time. Here you see the hepatocyte that is half dead almost, and this will be soon ready to release the parasite which are going to infect the red cell. However, at this point, even though the cell that is hosting this parasite looks pretty sick, it manages to present antigen to CD8 positive T cells, perhaps also to CD4 cells, and there is suspicion that may also be involved gamma delta T cell and NK T cell. However, the most robust effect against the infection are CD8 cells, or at least this is the best, it is the protective mechanism that we can work the best. And the other may be as good, however, we don't know how to work with them. Now, many years ago, the story of the CD8 cells against the liver stages began when using a rodent malaria system, which is Plasmodium joellii, and we found that mice which have been exposed to the parasite repeatedly, either live or attenuated sporozoite, are capable of inducing CD8 positive T cell response against this particular sequence of the CS protein of the sporozoite. This CS protein is expressed by the sporozoite when injected by the mosquitoes, and then goes with the parasite up to the liver stages, and still there is some residual expression in hepatocyte, and this epitope is presented in the surface of the hepatocyte, and these cells not only are induced after repeated exposure, but they are also capable of protecting. So these cells, specific for this epitope, and there may be CD8 cells against a lot of other epitopes from a lot of other antigens, we just don't know which are they. They can be induced by immunization with parasite, attenuated or live parasite. These cells have the capacity to inhibit parasite development to a significant extent, up to a point almost of providing sterile immunity. And most important, immunization with subunit vaccines, which can be attenuated viruses, particularly attenuated viruses. A lot of work was done many years ago in my and other laboratory, which showed that when we immunize with subunit vaccine that contain this particular sequence, we induce a lot of CD8 cells specific for this sequence, and in fact, mice can be protected against infection when they have enough amount of the CD8 cells. That's indicating that CD8 cells, together with other mechanisms of protection, are important for resistance to infection in mice and probably also in humans. Now, what we have learned about these CD8 responses through the years, and I just go simply showing it, that this CD8 response is extremely fast at developing. We can see CD8 responses as soon as 2 days, and peaking at 4 days after the mice are exposed to the parasite. This CD8 response reaches a fantastic peak very quickly, incredible rate of proliferation, and as this expansion phase reaches a peak in here, very soon, it could be between day 4 or 7, then it contracts very rapidly. Most of these cells which were created here died in here. I assume that the idea for the immune system is to produce a lot of CD8 cells, they do the job, and then the cells which have done the job die, and what we're left is with a few cells which are memory cells that last for several months. We know that this response, as I said before, is extremely fast. 24 hours after immunization, in fact, we can find it. And more recently, we can see that 8 hours after exposure to parasite, we can see dendritic cells, which are involved in the induction of the CD8 cells in the draining lymph node of skins. It is strictly dependent on dendritic cells. It is strictly dependent on CD4 cells. If CD4 cells are not available, and CD4 cells that produce IL-4, this CD8 response does not develop. And the protective memory cell, which is induced under this condition, is basically a peripheral effector memory cell, and that makes sense because those that actually kill the parasite are in the liver, not in the lymph node. Therefore, this is the subset that matters when it comes to protection. More recently, we have been studying how is it that these CD8 cells are actually induced when mice or humans supposedly are exposed to parasite. And we learned, surprisingly, only recently, that the skin plays a very important role. The parasites are injected by the mosquito into the skin, and the percent of these parasites that are in the skin, they can be there for several hours, moving in the skin, looking for a blood vessel. Some of them are capable of finding a blood vessel. They go through the endothelial cells, and they infect the liver. This may be half of the inoculum in here. Another percent of the parasites which are injected in the skin, either by syringe or by mosquito, actually goes to the lymph node as if they find the wrong way, the wrong vessel, they get into lymphatic vessel, and around 20% of the parasite actually end up in the lymph node, and you just can go see it in the lymph node, in fact, swimming, until suddenly they are picked up by macrophage and dendritic cells, and they are chewed up, and then at this moment, it is in this lymph node where the parasite have arrived that the dendritic cells pick up antigen or pick up the parasite and induce the CD8 response, which we see later on. 2 days or 1 day and a half after this parasite goes into the lymph node, these CD8 cells egress the lymph node and they travel to the liver where they become resident memory cells. So it's here, so we know that they're induced in the skin. We know that after they're induced in the lymph node, they migrate to the liver. And then we learn also that the recognition in the liver is directly, or we believe is directly into the hepatocytes. This memory that is created here strongly depends on antigen persistence. Sorry about that, this is misspelled. But surprisingly, this parasite, even though infect and a single immunization, the antigen of this parasite, which is essentially dead, the antigen keeps being presented in the lymph node and also part in the liver for around 2 months. And this antigen persistence appears to be critical for the development of very strong memory CD8 cells. Other details with regard to the presentation also is interesting that it's pretty much a typical cross-presentation in dendritic cell, but I want to go in those kind of details. Now, how do we look at this effect of CD8 cells in experimental setting? What we see is, what we usually do is we can inject CD8 cells, which are parasite specific. And after injection of the CD8 cells, we can inject parasites which are live, that are capable of producing infection. Or we can actually just change the protocol and then inject the parasite first and then inject the CD8 cells. And what we see, the way we measure, is the parasite load in the liver by a real-time PCR based on the detection of ribosomal RNA of Plasmodium. And you can see here, if we inject parasite with mice with parasites, and we just leave the parasites there to develop, this is more or less the amount of parasite RNA, ribosomal RNA that we detect in a normal or naive mice, 10 to the 7. However, if these mice, after being challenged with parasite, later on we inject parasite-specific CD8 cells, we see that 3 hours after infection of the mice, if we inject the cell, we see a significant effect in parasite development, 2 to 3 log difference. And this is quite a significant effect, considering that this is a single cell that recognizes only a single epitope. You can imagine how would be the effect if you have several cells or many other cells that may recognize 1,000 or 2,000 of different epitopes. Obviously, one would expect that this could be essentially a sterile immunity. And we assume that therefore that the CD8 cell is in the liver, have gone to the liver, and then waiting there and identify and then kill the infected hepatocytes. Now, we know that one important aspect of this effect of the CD8 cell is that after being induced in the lymph node, and around 2 days after priming, these cells in the lymph node acquire the capacity to egress, and then they can go into the spleen, to the lung, and importantly in the liver. And it's in the liver where these memory cells, which memory cells are called as such after 25 days or up to 30 days after immunization, because they acquire the characteristic of memory cell, they go and sit into the lymph node. Now, we have been interested in trying to understand what are the functional and genetic features of this cell. What is unique about these cells which decide to live in the liver and not in the spleen, for instance? The first thing is the functional properties. Here we have the way we do the experiment is just simply we inject CD8s, which are specific for the parasite. This is a T cell receptor transgenic cell, which are injected into mice. Then we immunize the mice with the parasite, and we wait 30 to 45 days, and we pick up the spleen, and we pick up the liver, and see how similar are they. The cells in the liver and the cells of in the spleen are identical, are exactly the same one we injected, because they have the markers that allow us to identify, and they are genetically identical. They have exactly the same T cell receptor. The only difference is that some cells decide to have a house in spleen, and the other cells decide to have a house in the liver, and they live in there for reasons that we don't quite know. First, first analysis we do is, are they functionally similar, the cells which decide to live in the liver versus those that decide to live in the spleen? And for this purpose, we just do a polyfunctional analysis in which we are able to determine at the single cell level what are these cells producing. So for instance, we have here the percent of cells which are capable of having cytotoxicity, CD107A, which are also capable of producing interferon gamma, capable of producing interferon alpha, and capable of producing IL-2. And here we have other populations which are capable of doing cytotoxic activity alone or interferon gamma alone. So we have polyfunctional cells which can do a lot of things at the same time after immunization, and we have monofunctional cells which just do a single thing after immunization. So when we compare the different memory setups in the different organs, this is what we see in the case of lymph nodes. We see that many of them, or the majority of the memory cells which are in the lymph node, can be classified as polyfunctional cells because they sort of can do everything. They can be cytotoxic, produce interferon gamma, TNF, as well as IL-2. Very few of them are monofunctional, and even though it doesn't matter at the time, this doesn't change very dramatically according to the time point at which we pick up these memory cells. However, when we compare the memory cells in the liver, which again are the same cell, they are the sisters of the cells in the spleen, they are genetically identical, what we see is that once the cells have decided to live in the liver, they adopted a different attitude, and they are very monofunctional. They do not produce much cytokine, and they are, if anything, mostly almost exclusively cytotoxic. So these cells, although they are identical, once they go to the liver, somehow undergo a differentiation process which is completely different for reasons that the liver is a unique organ and does a lot of unique things. Clearly, in this case, what it's doing is influencing the memory cells which live in there, and they made them hyperfunctional and keeping mostly their cytotoxic activity. The reason for this, we don't know. It could be an attempt to immunosuppress the response, or it could be an adaptation of the CD8 cell so that they can have a single function and not produce too much cytokines in the liver, because that could be deleterious for the organ, of course. We don't know exactly the answer, but we know that the cells undergo a differentiation which is unique, and this is an organ-specific differentiation. The cells in the spleen are not the same in the liver, probably they are not the same in the lung, and probably they are not the same in the brain. Now, functionally they are different. What else is different? Since we have this good setup, we are able to isolate spleen cells, memory spleen cells, and we are able to isolate liver memory cells, and we can isolate it to an extent which is pretty pure after a column enrichment by magnetic cell sorting followed by cell sorting. So we can have populations of spleen cells which can be as rich as, this is liver at 98%, or memory spleen, this is liver, which is also highly enriched, is almost pure. And that allows us to take a look at what's going on with the transcriptional profile of this particular cell population. And the surprise is that, again, because they were functionally different, what we see here in transcriptional profile, that memory cells versus spleen cells, both are memory compared to naive cells, they have a dramatically different transcriptional profile, and they look like we were comparing cell types which are completely different one from the other. That tells you that not only there is an influence in the functional profile, but obviously the fact that these cells decide to live in the liver rather than the spleen influences dramatically the transcriptional profile of the cells, and it's a differentiation which is unique, which you don't see in any other place. But you see here, many of these chemokines have upregulations as well as downregulations of molecules which are of interest from the viewpoint of immunology, and the PCR, real-time PCR of some chosen gene versus the result of microarray, pretty much are very concordant. This meaning that our microarrays are fairly decent. As you can see, interesting genes are CD40 ligand, CXCR3, MMP9, which are proteins which are involved in different processes of migration as well as functional activity. Here again, it's just a list of different genes which are up or down, but I'm not going to get into this, and I'm not sure if I want to get in. There are too many genes. There are around 300 genes which are different between memory cells in the liver versus memory cells in the spleen. And you here have more of a prominent, there are significant differences, cell cycle genes. Those cells which are in the liver, for reasons we don't know, they like to proliferate like crazy. They're continuously proliferating. I don't know why. They do express higher amount of certain effector molecules such as gram-sine B, FAS. They have a much more down-regulated transcriptional profile for transcription factor and some intracellular signaling. We're not going to go into detail of what is what. The bottom line is what is important here is that CD8 cells are not equal to CD8 cells equal to CD8 cells. The CD8 cells differentiate depending upon where they go. If they go to the brain, they will adopt a particular characteristic. If they go to the liver, they adopt a different characteristic. If they decide to live in lymphoid organ, is different. And it kind of makes sense because those cells live in different environments, and therefore the requirements of their function will be different, and that's why they just simply undergo unique differentiation, which is very much specific for the organ where they live. Now, we know that these CD8 cells, besides being unique and unusual, and they live in the liver, and they can live in the liver for 6 months, in mice at least. We can detect them for 6 months, they are still there hanging on. We can purify them, and the frequency of the cell is very similar. Now, when we talk about the cell, we usually think about CD8 cells in these terms, in cartoon terms. The CD8 cells come here, recognize the antigen, they produce stuff, and the cell gets killed, and then the CD8 cell go to another one, kill another cell, and so on forth. Now, the question is that we said, is this really the way it happens in vivo? Is this cartoon really the way it happens? We know that CD8 cells kill, but do they kill just by going one cell, sitting there, killing, and then going to another? Or is it a little bit more complex process that occurs in the liver at the time of infection? For this purpose, we have done imaging of to study the behavior of antigen-specific CD8 cells in real time. First, I will show you some static imaging of the CD8 cells, and what we do here is just simply immunize with parasites. We wait a little bit more. We can wait weeks or days, and then we challenge with live parasites which contain GFP. Before the experiment, we inject here with anti-CD8 cells to find and the mouse is alive. This is a full mouse here, anesthetized. Surgery has been done to expose the liver, so they have the liver exposed, and the objective of the microscope is here. We just keep the mouse alive for around 5 hours. And you can see that the mouse is here in the microscope, and then you just can go see it. I think here is the mouse. And then you just can do, this is confocal microscopy that allows to do this liver surface imaging. What we see here is just with the response, which is the endogenous, the first finding was that these few hours after injection of the parasite, this is the parasite, the green thing, and the red things are CD8 cells. It's the fact that CD8 cells appear to congregate and they just sort of rush to the site where the parasite is. You see there. Now, when we look at a little bit more in detail and we compare immune mice versus naive mice, here you see some parasites that grow, and there are some CD8 cells in there, but more than half of the parasites in immune mice are associated with CD8 cells which begin to surround them. The CD8 cells surround the infected hepatocyte and form a cluster around there. And you see here that actually the parasite is kind of being destroyed. It's in pretty bad shape. This clustering around the infected hepatocyte is very seldom seen when we do the same with naive mice. With naive mice, the parasite grows, and very rarely we can see a CD8 cell that appears around there looking about. This is the prevalent image that we see, CD8 cells which surround the infected hepatocyte. And now this here is the quantification of the process. Here you have naive cells, We define a ratio to define cluster. You see, in general, we see very little or no cells which are clustered around the infected hepatocyte in the case of naive mice, but in the case of immunized mice, where you see here that most of the parasites are associated, and sometimes with as many as 15, 16 CD8 cells which surround the infected hepatocyte. So this is the common theme. Once there is immunization or there are CD8 cells around, they rush to the site of infection and they begin surrounding the infected hepatocyte. Now, to take a better look at what's happening, we also did dynamic imaging, and we essentially use basically the same, except that we modify a little bit the protocol in which we first, these mice are naive, they inject the mice with a GFP parasite that produce GFP so we can see the green thing, and then we inject after that labeled CD8 cells which we know are specific for the antigen that is expressed by hepatocytes. 5 hours after these mice have received the CD8 cells which are previously activated, these are memory cells, we inject the memory cells and then we take a look at the mice, and what we see is very similar to what we used to see in the case of the endogenous response. These red cells are the ones that we injected, and you see the parasite in there. That is no, okay, well, it doesn't work, doesn't matter. You see the cell, this is the parasite, and you see that after 5 hours that we inject, CD8 cells begin to congregate and they begin to surround the infected hepatocyte. I have this impression of kind of in the West, Wild West, where you have the peregrine. I don't remember these people that used to go to there in these wagons, and the Indians are surrounding and this is what the fact what you see in there. And what is important is that this clustering around the CD8 cell, it is strictly antigen specific. If we infect the mice with Plasmodium berghei, which contain an epitope which is slightly different to the Plasmodium joellii, and the cells which are specific against this do not recognize this other thing, the cells which are specific for joellii can care less about a parasite infected with berghei. They are capable of seeing that this is not the parasite that they are capable of recognizing. But if you have the joellii parasite and the anti-joellii CD8 cell, is when you see the clustering around, and this is shown in this particular case, when you see clustering of CD8 cells when the mice are infected with berghei, wrong epitope, or with Plasmodium joellii, which is the right epitope, as you just can see in there. And we can do also other studies. So we know that the cells cluster and they approach the infected hepatocyte. By using the dynamic imaging, we can actually see what happens when the cells begin to approach the infected hepatocyte and what happens with them. They just go around or not. Usually, when you're seeing this T cell, they are going very fast. Screen like scanning the liver. They go back and forth, back and forth, back and forth. However, the behavior of these cells, which are continuously like crazy around the liver, when there is an infected hepatocyte, they cannot change. And what you see here is, for instance, here we compare the speed of the CD8 cells when they are inside the cluster versus outside the cluster. What you see here, outside the cluster, the cells in general go very quickly. They have a high speed, scanning the liver. However, when they find the cluster and they get inside the cluster, they move very slow. They just get trapped there. They don't move exactly the same. And when we look at the displacement rate, I mean the space that they cover during a certain time point, the same thing happens. Cells which are inside the cluster or within the area of cluster are very slow. They slow down. It looks like the whole environment around the infected hepatocyte is sticky. So the CD8 cell goes there and gets stuck and then move very slowly and cannot get anywhere there. And then eventually they could leave. If they leave the cluster, then they reacquire speed again. So there is an environment in the infected hepatocyte that almost trap the CD8 cell, and this entrapment we don't know what are the molecular basis, is antigen specific, because if the cell is CD8 against another epitope, they couldn't care less. And there are other changes like meandering index, which is the kind of cell when they do loops and stuff like that. When they are inside the cluster, they are behave much better, they barely move, and they just sort of stuck in that particular region. Now, if you excuse me, I want to show you now the actual videos of what happened in the liver in real time. We observe different phenotypes of this when we do dynamic imaging of this parasite. I hope it works. All right. Here you have, this is mouse which is alive. Here you have the hepatocyte, and here you see that it's being surrounded by C. You see, suddenly the GFP disappear, and the cells essentially, we believe, the obvious interpretation here, I'm going to show it again, is that these cells, which were around here, killed the hepatocyte as the GFP have disappeared, and this is around 5 hours after injection, after an observation of around 25 minutes. So this is one type of phenotype that you see the disappearance of the GFP. You have another type of death that we believe they are, which we call it phenotype 2. This is very slow. You barely can see, this is the infected hepatocyte, and you can barely see that the GFP begins to decrease very, very slowly, and you don't see much of a very dramatic effect. We're going to show it again. You look at the beginning, and then GFP begins to go down, down, down. It is not that the parasite changed the focus. The brightness of the CD8 cell is the same, and this begins to disappear. This is another kind of phenotype. It's a slow death. It's like a different mechanism. And finally, we observe other type of death, which we call, which is the most kind of dramatic, probably. We call the phenotype 3. Here you have the one. You have cells which are around, and look at this. It's like it's like a shark. See, the CD8 cells are around here, and they begin to destroy the infected hepatocyte and the cells. You have the pieces of GFP flying all over, and it's not that the parasite is leaving the cell. The parasite is not capable to do anything. This is 20 hours after infection, and the parasite can leave the hepatocyte all around 40 hours. So clearly, what is going on here is definitely what appears to be destruction of the parasite induced. Here there are less cells, but you see pieces of, and these parasites which have been living here, they essentially die because they are not capable of undergoing any differentiation or invasion of erythrocyte, which is their next step. Now, and we can quantify this, as a matter of fact. If you look at this, at the fluorescence, you just choose the background, you just see the fluorescence of the parasite in naive mice. This is a 5-hour observation approximately, and then you see that the fluorescence in this case remain pretty much the same. When you look at that, this is background, and this is the fluorescence of the parasite. When we look at mice which have cells, then we see this different phenotype. Sometimes you see sudden disappearance of the parasite in spite of the fact that CD8 cells are there. You see the fluorescence, boom, disappear, 96% minus, and essentially this is kind of violent death. The different series, 5 minutes after the other. The microscope every 5 minutes revisit the same parasite. They have other kind of death which are not as fast, like this and this, which for it lingers there, still around, and the parasite begins to just kind of disappear only little by little. And then we have this other parasite that I showed you before that appears to involve the destruction of the parasite, and pieces of GFP material appear to be induced. Now, this behavior of CD8 cell is important. It is mediated by molecules. We don't know which molecules they are, but we know that they are, because if we treat the CD8 cells with pertussis toxin before injecting into the mice, what we see is that the cells lose the capacity to cluster, even though they go to the liver in equal way. They do not cluster. Here is the normal CD8 cells, and these are cells which have been treated with pertussis toxin. and they lose the capacity to cluster, and they also lose the capacity to kill the parasite. Here you have the normal parasite load in naive mice, parasite load in naive mice which receive CD8 cells, and this is the parasite load of mice that receive CD8 cells treated with pertussis toxin. As you can see, this treatment, which do not kill the cell, which do not change the capacity to live in the mouse, and after treatment with this drug, the parasite do no longer have the capacity to find and or kill the parasite, indicating that they are involved. The only problem that G protein coupled receptor is a large family of receptors, and there are 100 or more, and we really don't know how many they are. But clearly, this is a directed process in which important molecule families are involved in here. So what we see with regard to the visualizing the effect of CD8 response, in fact, we believe that this is the first observation of a pathogen destruction by the CD8 cells. There are studies in tumor, but actually in vivo there are not studies which show destruction of pathogens. What we find is that there are clusters of multiple CD8 cells around the infected hepatocyte, and this appears to be mediated by hepatocyte changes in there. Something is happening in the environment of the infected hepatocyte that makes it sticky or attract the CD8 cells, so they go there, and you can see 10 or more than 10 or 15 cells which surround the hepatocyte, and eventually one of these cells are going to kill the parasite. We don't know which one. That is what we see more often. You can see sometimes a single cell and a single parasite, and there are different phenotypes, as I showed you before. There is slow death, and we have dramatic death, and we believe that possibly that indicates that different mechanisms used by the CD8 cells are the ones that are operating in this case. We believe that the CD8 killing it is a prolonged process. It's just not always a rapid event. Most of the video I show you takes after 5 hours. We have injected the cell, and they take 1 hour or 30 minutes before the cells move around. They move around the infected hepatocyte. They sort of toy with the hepatocyte until sometime one go there and kill the hepatocyte. We don't know exactly how is that. Finally, it's just showing the people that actually do the work. Most of the work have been done by Ian Carman, a British postdoc that was in my laboratory for many years. He's a fantastic guy, have done a lot of work and have been able to advance a lot of the field in collaboration with our colleagues at Institut Pasteur, Jové Menard and Rogerio Mino, and some other of my students. And thank you so much for your attention.